Hey guys and gals, this video is going to have three goals for you. First, I want you to calculate the probability of traits appearing in an organism. Second, I want you to describe organisms in terms of heterozygous and homozygous. And finally, I want you to distinguish between phenotype and genotype. So, first and foremost, probability. Well, what is it? Probability is the likelihood an event will occur. So, if we think of an example here, if you flip a coin, you have a 50% chance of getting heads. But if you flip the coin again, you have a 50% chance of getting tails. So probability is a chance that something is going to occur. And each attempt is going to have a different probability if you're asking for, like, how many times of getting head 3 times in a row. Well, it would be like 1/2 times 1/2 times 1/2, which is equal to 1/8. So probability is going to be talked a little bit about this because basically what we're doing is probability. We're trying to find out what is the probability of getting a certain trait. So to get into that idea, we have to talk about segregation. Segregation is going to predict what kind of outcome you're going to get. So as you guys are probably aware, you have two Ys. If you have two alleles for yellow, the gametes that are going to be produced here are going to be big Y and big Y. If you have a green pea that has little y, well then the gametes that are produced here are going to be little y, little y. And for review, the gamete is just going to be a sex cell, so think a sperm cell or an egg cell. Well, because we know that alleles are going to separate into gametes, so you have a big Y and a big Y, it's going to separate into its two parts. Each gamete gets one allele. When the gametes come together, the new cell will have two alleles again. So for example, if this cell here and this cell here came together, then the new cell would have a big Y and a small y. So here's sort of how this works. Alleles are going to separate, which we know from the law of segregation, at which point in time each sperm and egg cell are going to have one allele. When fertilized, the alleles come together and make another allele. What this will do for us is, if you see here, which is called a Punnett Square. So you have the girl here and the guy here. The girl has big T, little t. Well, because of segregation over here, the big T and little t are separate into a big T and little t. Now these right here are just showing you egg cells. And these over here, because they come from the guy, are showing you sperm cells. And this is showing you what will happen. So if a sperm here and the egg here come together, they're going to form big T, big T. If the big T right here comes together with the male small t, then we're going to have big T, little t. And if we have little t here coming together with little t here, we're going to have little t right here. This right here is called a Punnett Square. This whole thing is how biologists try to predict what is going to happen when a male and a female come together and reproduce. So in order to actually get into this though, we have to have some key terms. There are 4 words here you have to know in order to understand what's going on. The first one is called heterozygous. Heterozygous just means that an organism has 2 different alleles for a trait. So in this case here, they're going to have big T, little t. That would be heterozygous. The other word would be homozygous. In this case, this is when an organism has alleles for the same trait. So an example here would be big T, or little t, little t. Now, some examples to play with right here comes from weird pig monster looking thing. In this case here, if you have a big B and a big B, we call you homozygous. But since they're both big, we call it homozygous dominant. In this case here, we just call it heterozygous, because heterozygous is going to mean you're going to have big B, little b. Now over here, we're going to call it homozygous again, but rather than say homozygous dominant, we're going to call it homozygous recessive. And that's how you can do a lot of this stuff, is if you hear homozygous recessive, well then you know they're going to have the two smaller letters. Now, there's a real trick here that's going to be potentially a problem for people on exams. And that is that you can choose whatever you want for the letter of an allele. You can use B, you can use C. You'll find a lot of textbooks, and you get to college, they stop using letters and start using like abbreviations. So rather than having like tongue rolling, they might do TL1 versus TL2, where 1 would be the dominant trait and the 2 would be the recessive trait. Again, they're just changing it up. This is still the same as the big letter, this is still the same as the small letter. They're just trying to make it confusing, and, well, frankly, they're trying to make it look more scientific than having big B versus little b. It's better to have, hey, it's TL1 versus TL2. That sounds more intelligent, and it probably sounds more scientific, which is probably why people are doing it. Some other terminology is the term genotype. Genotype is the alleles that an organism has. The phenotype is the actual appearance. There's a situation here is that, now before that, let's go ahead and pull this up because this actually shows you a better example of what I'm talking about. So the phenotype is what you actually show. So if your phenotype is blue eyes, then you have blue eyes. The genotype is what alleles you have. Well, since it's blue eyes and blue eyes is recessive, well, that means that he has to have little b, little b. Now, the phenotype here is going to be brown eyes. Now, brown eyes is dominant. What that means is that he could have the genotype big B, little b, or big B, big B. We just don't know. And the problem here is because dominant traits can hide the genotype. If you have brown eyes, you don't know what your alleles are because the big B is going to block out the little one. You have no idea what the second one is. You could have big B question mark because you just don't know what that second one is going to be. So, but if you're recessive, you know for sure what the genotype is because recessive, you have to have both small letters in order to have it. So, Punnett squares. I explained all this a minute ago. I'm not going to go into too much more detail, except I'm going to explain one more thing. With Punnett squares, it's tradition that the female goes on this side right here, and the male goes here. So female on the left and male on the top. There's no real reason for this, but it just comes down to it's better to have a rule for this. That way if you see someone's Punnett square, you know for sure that the female is over here and the male is over here. So here's a quick sample problem for you guys. So a woman who is homozygous recessive for Huntington's disease is married to a man who is heterozygous for the condition. If Huntington's disease is dominant, what is the probability their child will have the disease? Well, the first thing you want to do is you want to find out what the alleles are going to be. And the alleles here are going to be little h for the mom because the mom has already been said that she is homozygous recessive. So we know that she has little h. Now, the dad is said to be heterozygous, so you know he has to be big H, little h. Once you have that, segregation is going to occur. Then, these here are the alleles inside of the eggs. This is going to tell you what alleles will be in the eggs. Well, the mom only has a little h and a little h. The dad has a big H and a little h. So what that means is that the mom will have little h, and the dad will have big H, little h. And you can probably guess where I'm recording this video because you just heard that bell sound. The second thing here is once you have that, you're going to put it onto a Punnett square. And this is my little Excel version of a Punnett square. What's happening here is you have the father up here, and his father is big H, little h, so the big H goes here, the little h goes here, and here you have the mom who's little h and little h, and here are their possible babies. Well, they have two of the babies will be big H, little h, and two of the babies will be little h, little h. What this means is that the baby has a 50% chance of having Huntington's disease. And this is how a Punnett square works. It's just a way of easily predicting what's going to happen. Now, Punnett Squares come in a more advanced version called a dihybrid cross. Dihybrid crosses are only used when you are trying to predict where something is on the chromosome. You can use these dihybrid crosses to find out where genes are located. However, for the course of this class, we don't actually do that. That's going to be more of an advanced placement. So if you do take AP Biology next year, you're going to be doing a lot more with these. But I still want you to know how they work, because if you do take AP Biology, I want this to become familiar so you actually know what's going on. So what happens with this is that basically it's for two traits. So you have the Y trait and the R trait. So Y here would stand for yellow, and R here would stand for rough. In this case, Y is dominant and big R is dominant. So smooth is dominant and yellow is dominant. Wrinkled is recessive and green is recessive for P's. So if you made these two P's here have a baby, well, their offspring would be big Y, big R, little r. And that's because what's happening here is it's impossible for any other combinations to occur, because the parents only have one allele to give. But if you made these babies have babies, what's going to happen is you're going to have a female that's going to be big Y, little r, little r, and a male that's big Y, big R, little r. So you need to make the gametes. Because of the law of segregation, these gametes are going to separate independently. So big Y, big R becomes one possibility, and that goes right here. The big Y, little r is another possibility, and that one's going to go right here. Another possibility is little y, big R, and that's going to go right here. The final possibility is little y, little r, and that one's going to go right here. And the male has the exact same thing going on. So basically, you just do what you would like do in math. You distribute, and you find out all 4 possible combination of gametes. Then you put them onto a Punnett square, and you get something that looks like this. And there are 16 possible phenotypes, because as you can see, there are 16 different squares here, and there are 16 different genotypes here. So there's YYRR, YYR, little r, as such. So for problems like this, what you want to do is break it apart into its simpler parts. So a man who was homozygous recessive for tongue rolling and albinism had a child with a woman who was heterozygous for both of them. What is the probability their child will be an albino tongue roller? Well, same thing as before. Find out the man's alleles, and he's going to be little t, little a. Find out the woman's alleles, because he's going to be big T, big A, little a. Then you have segregation. Here are your possible gametes. Well, the man's only going to have t, little a, because he only has those 2 alleles. He can't give anything else. The woman, though, is going to have 4 possible alleles. She's going to have big T, big A. She's going to have big T, little t, big A, and little t, little a. There's 4 possibilities with her. And once you have this, you just put it onto a Punnett square. So you just have T and A over here, and you just put the other alleles on the other side right here. And your Punnett square would actually only have 4 boxes if you were to draw this out, because you only have 4 possibilities here and 1 here. The last idea here I'm going to talk about is called independent assortment. This is the idea that when Mendel crossed 2 heterozygotes together, he found out there was a phenotype ratio of 9:3:3:1. So if you go back to this chart right here, what you see is that 9 of these are going to have one phenotype, 3 of them are going to have another phenotype, another 3 of them are going to have another phenotype, and then 1 of them is going to be totally recessive. Now you're probably asking the question, why do we care about 9:3:3:1 as a ratio? Well, the answer, unfortunately, is you have to wait for AP Biology. But what I can tell you is this. 9:3:3:1 tells us something. It tells us that from a standpoint of math, that means when the alleles are separating, they don't affect one another. So the Y allele does not affect the R allele in the previous example. They're independent of each other. This is called independent assortment. So if the ratio was something like 9422, this would mean that the alleles were somehow affecting each other, and we don't know how. And actually, what you're going to learn if you take AP biology, you're going to learn how to take this ratio right here, or other ratios, and use it to predict where genes are located within the body. It's a pretty cool little trick you can do. And that's it, guys. I hope this wasn't too long of a lesson or too confusing. Have an awesome day, and I will see you all in class.